Hello, my name is Nicole Cueli, and I'm a second-year medical student at OSU CHS. My project I'm going to be presenting on today is about the sex differences in protein excretion in mice consuming a high-protein diet. Some background about this project is that normally the renal excretion of protein, also known as proteinuria, is absent or very small. Ingesting high-protein diets can elevate proteinuria and in the long term, increase the work on the kidneys by increasing glomerular filtration and higher energy requirement to handle the protein. Sex differences in renal function are well known, and thus differences in proteinuria may exist. The purpose of this study was to determine if sex differences exist in proteinuria in mice consuming a high-protein diet and investigate the potential roles of these sex steroids of estrogen and testosterone on the protein excretion. The methods used for this study were that healthy 3 to 4 week old male and female intact and gonadectomized mice were used. These mice were placed in individual metabolic chambers where the urine of each mouse could be collected and measured for protein concentration. The mice consumed a 14% casein protein diet for 25 days. A normal diet of protein consists of about 20%. Some gonadectomized female mice received exogenous estrogen and gonadectomized male mice received exogenous testosterone. Proteinuria was then measured via dipstick measurement in protein excretion. The results are shown. In figure 2, we looked at protein excretion in intact and gonadectomized mice. In the females, we saw no significant differences between the intact and gonadectomized groups. However, in the males, there was a significant difference between the intact and castrated mice. In figure 3, we look at effects of the sex steroids on protein excretion. In the females, there were no differences or no significant differences noticed. However, in male mice, the intact and castrated mice with testosterone showed a greater protein excretion. In the fourth figure, once the project was concluded, one mice was then killed and we measured their kidney weight to body weight. In the female mice, we did not see any difference. However, in the male mice, we saw that those with added testosterone had hypertrophied kidneys. So they had a higher kidney weight to body weight ratio. In conclusion, the results of this study suggest that the male sex steroid induces high proteinuria in mice consuming high protein levels. The female sex steroid plays no role or only a minor role in proteinuria under these experimental conditions. Food intake data not shown in these results was not significantly different between males and females or among the intact and gonadectomized mice. Thus, the protein excretion data in this study appeared to be produced by the specific actions at the kidney. Testosterone causes kidney hypertrophy, which might be directly or indirectly responsible for the higher protein excretion. Our results suggest that androgens may account for the higher incidence of kidney disease in males compared to age-matched premenopausal females. My citations, and thank you very much.